So today, we do have a lab quiz. We'll start the unknowns today. We'll finish exercise 20 today. Yes. And then next week in lecture, we'll be finishing off module 10. So we're starting today, finishing it off next week. We do have a quiz on Monday. That will be on module 9 and where we get to today, module 10. Okay, so touch on the intro to module 10. You do have 2 quizzes next week also, so each day has a quiz. The following Monday is the Unit 3 exam. Okay, so that's on the 17th. We got about a week and a half before that approaches. That exam course will be module 8, 9, and 10. Let's see here. That should cover everything on her up and coming schedule. You have already, I mentioned on Monday, and so you should, and hopefully you should have seen the Frontline Documentary bonus point assignment. That is an additional assignment above and beyond the unit bonus point assignment, okay? Both of which are worth 5 points each. So, you know, just like we've had each unit so far, there are questions you answer, you turn in, you know, get them right, you get the 5 points. The same thing, of course, with this, you know, a couple questions to be answered after watching that Frontline video. And then you submit, and hopefully you get those, as long as the answer is correct, you get those points based upon, you know, correctness. So module, I should have had something to drink for water, but module 10 is, we're gonna be looking at infectious processes and epidemiology. So the process of an infection moving into a disease state. And then epidemiology is the study of that process. The material on Canvas for that, you know, the textbook for you to read, you know, it displays it a little bit differently than I'm used to. The progression of it displays it a little bit differently than I'm used to. So I've tried to organize it in a somewhat, you know, complete fashion, but that follows what you guys will be looking at. That being said, you know, you have your learning modules. You should be able to answer. There's 10 of them for this one, so this is kind of a biggie one as far as the amount of material that you're looking at. As you actually are looking at those, doing those readings and those videos, you're going to see that is the case. Which is why I have 3 days worth for it, the discussion of. All right, so the first learning objective, of course, is to classify symbiotic relationships. And I think we've talked a little bit about that idea of symbiotic relationships. This is actually covered in the chapter 4, the first item, the first item for your reading. And I will remind you, I made mention of it on Monday, but I'll remind you now. For that, I'm really interested in you reading 4.1 only. Okay? You do not need to go into the other sections of that chapter. Interesting material. It goes into a little more detail specifically of microorganisms, but we're going to be focusing on that first segment, okay? Now, the symbiotic relationships. Symbiotic relationships are required relationships between 2 groups of organisms, okay? And then we have our non-symbiotic relationships. The non-symbiotic relationships are when 2 organisms are living together in the same environment, but it isn't required for either, okay? They have 2 terms in this textbook that I will tell you right now I won't use. One is neutralism for a symbiotic relationship, where that symbiotic relationship, they say, is where neither benefits. I don't think that that's a symbiotic relationship personally, right? Because in a symbiotic relationship, a required relationship, they must both be present. There has to be some sort of benefit for one or the other. I noticed that their definition of symbiotic relationship does not state that it's a requirement, okay? So they don't even bring up really this non-symbiotic relationship where it's not required. We can have two groups of organisms living together that, you know, do well together, but don't actually require the other one to be survive. And then, but the symbiotic relationships are ones in which you do have that requirement. The other term that they have, I'll tell you right now, that you'll notice is not on my list here also for symbiotic relationships, would be amensalism. And I don't like the use of that term solely because I have yet to see a textbook describe that relationship properly. They always, for amensalism, they describe it as one of the parties is harmed by the relationship, and the other party is indifferent. Okay, neither benefits nor harmed. Okay, and that first, the description they always give for this is a microorganism producing an antibiotic. Okay, in that environment, that antibiotic is harming another organism. So the other organism is being harmed while that particular microorganism isn't benefiting or being harmed. And I disagree with that statement because they're killing off competition for a food source. So as far as I can tell, they are benefiting, okay? So in that relationship. And therefore, to me, based upon the definition of amensalism, that doesn't work, okay? That's, you know, my personal opinion, so I'll never use those terms on the test, okay? Just getting that out there right away. So the three that I would want you to recognize and know would be mutualism, commensalism, and parasitism. Now, mutualism is where they're both sharing, okay, and they're benefiting from that relationship. So an example of that would be bacteria in the gut, right? We're providing a nice warm environment and a continual food source. They're helping us break down that food source and into nutrients that our body can absorb. We benefit, the bacteria benefit, okay? It's a mutualistic relationship. Commensalism is where one party benefits and the other is indifferent, okay? And in this case, an example might be something like mites in human hair follicles, okay? We don't benefit, we're indifferent. We don't care. They're there. They're not harming us, right? But we are providing a home with warmth, okay, and for these mites. And they're there. They're part of our normal flora. And then parasitism is the last of those symbiotic relationships where one party benefits, the other is harmed. Okay, and so like I said, to me, a bacteria that produces an antibiotic is in a way falling in that category, right? They are benefiting because they're eliminating their other food source. However, that being said, you know, when we think of parasitism, we're usually thinking of intracellular parasites, right? Like viruses, or we can think of, say, their example they use is tuberculosis in the human lungs, right? Not all parasites are intracellular. Okay. Generally, we jump to that right away, but not all are. As long as they're in the body and they're doing damage to, and you know, they're benefiting from that environment, and the other organism is being harmed. That is a parasitic relationship. Now, when we go to non-symbiotic relationships, again, these are relationships that are not required, right? That the two microorganisms do not need to be together. They can grow without the other one present. And so that's where I say, kind of with the description that they use for neutralism, I'd say that's more like a non-symbiotic relationship. Neither benefit from it take one away. Since there's no benefit, you take one away, it's not a requirement. The other one can survive. That being said, there are non-symbiotic relationships that can be classified into additional categories, and that is a synergistic relationship or an antagonistic relationship. And a synergistic relationship is, though they're not required to be together, when they are together, they grow better as a pair than not, okay? So like we can already, probably in our lives think of relationships, right, that could fall into synergistic relationships. Do they require each other? Did they, if they're not partnered up? No. They can still survive, they can still, but together they seem to thrive better. We can also probably find in our life individuals who go with an antagonistic relationship, where again, they don't have to be together, but when they are together, right, they do worse together than they do when they're individually separate, so they're living on their own. So those are categories within the non-symbiotic relationship. All right. Now, for us as people, of course, we've talked about the fact that we have organisms in and on the body, and these organisms colonize the body without causing disease, right? They're present without causing the disease, and these are referred to as normal microbiota, or you might hear the term. You might hear me throw the term normal flora. Because when I was going to school, back when, which wasn't that far back, plenty far back enough, that was the term at that time. Okay, the microbiota is a more current term. They moved in that direction. Normal flora is what kind of fell with when we were talking about plants, right, and the groups of plants that are out there. That was kind of the idea, I guess, where it originated from. But in any case, you might hear me flop back and forth. I try to stick with microbiota, but occasionally that other one slips out. It means the same thing. Now, for our normal microbiota, we have approximately 10 times more microorganisms in and on our body than we do have of our own human cells. Okay, so we have a lot of microorganisms. We are not sterile. Okay, we are a organism that maintains another population, right? And these population of microorganisms are found really on any tissue that is exposed to the outside world. So you're looking at the skin. That's the most obvious, right? So with the eyes, the portion exposed to the outside, yes. The internal, no. As far as the respiratory tract, the GI tract, we'll start with the GI tract. The GI tract from start to end is going to have normal flora, with the exclusion of internal organs, like say the pancreas or something, an actual organ that's inside. If it falls within the tract, the digestive tract that is part of the tubing going from one side to the other of the body, that all is going to have normal microbiota. Where we don't see, in the respiratory tract, it's more the upper. The lower respiratory tract, as you move into the lower respiratory tract, it becomes more of a sterile environment. Okay? Each of these areas is going to, that we find normal microbiota, are going to have their own population that is unique for that area, because each of these areas of the body create these little micro environments. The surface of the skin is a little saltier, even the nasal passageway is too, right? The stomach, you say, well, we don't have normal flora there. We do. Normal flora can exist in the stomach, and these are organisms that are going to be able to survive in the little more acidic environment of the stomach, right? The lower digestive tract is more anaerobic than the upper digestive tract. So depending upon where in the body, each of these environments is unique, and so you'll have your own groups of microorganisms found there. Where you do not normally find microbiota would be on any of your internal organs and bodily fluids. Now, when we talk about this normal microbiota too, we do actually group them into, the normal microbiota is actually grouped into two different classes. We have the resident microbiota and the transient microbiota. The resident microbiota are part of the normal microbiota throughout one's life. It's mostly a commensalism relationship. Mutualistic relationships are there too. And it is going to be found in the upper respiratory tract, the upper digestive tract, the lower digestive tract, the urinary tract, but specifically on the skin right outside and just the entrance of the urethra. Okay, that's about as far as we see it there. Reproductive tract, well, that would depend upon which sex we are talking about. For the male, it's the same thing as it'll be found just in the same areas as for the urinary tract. And for the females, it'll be found on all the outside surfaces of the reproductive, as well as the vagina. Other than that, you start moving into sterile regions. Skin, okay, has resident microbiota, generally found in the deeper tissues, because skin is constantly being sloughed off. So the resident microbiota tend to be on the deeper tissues. And then, as I mentioned before, on the eyes, you're looking at the surface of the eye that has contact with the outside. Okay, not as much on the portion of the eye that is inside of the skull or the inside of the eye. Transient microbiota is the microbiota that changes over time. And they can be some microbiota that we might have for short periods of time, and then we lose, right? And why? Well, first of all, before I get to why, where are they found? They're pretty much found everywhere that the resident microbiota are found, okay? It's just that they're not established as long-term. Why do they not persist in the body as long as the resident microbiota? Some of it is competition for the food sources and surface area for attachment purposes, right? So some of that might have to do just with the fact that you have the resident microbiota, so these are going to come in for short periods of time. They might try to establish, but they're not able to stay long. Other reasons is that our own body's defenses eliminate them. Okay, so that's kind of unit 4. We'll talk about our body's responses and a defense against these microorganisms. But that can come into play. Another reason is chemical or physical changes in the body. So as we go through puberty, there's changes in the body that are going to result in, like for women, pH changes in the reproductive organs, right? So you're going to see a shift occur in your microbiota for that area, right? Because that microenvironment has changed. And then later in life, when female goes through menopause, we see another shift. So those type of things can come into play. Others would be, say you're taking antibiotics. You know, you taking antibiotics can kill off you're going to be killing off some of your microbiota. They're going to be affected by this antibiotic too, depending upon the antibiotic and what its target is and what this microbiota is, right? So if you're targeting, using a penicillin base that's targeting mostly gram-positive, then your microbiota that are gram-positive are going to be affected.
Is that why antibiotics sometimes cause a yeast infection?
Yes, that has to do with the superinfection we talked about. Yeah. Because you're killing off your normal microbiota, and yeast is part of the normal microbiota. Keep in mind when I'm talking about this normal microbiota, it is going to include bacteria, it's going to include your yeasts, and protozoans. Okay? May include mites, right? So some of your other microorganisms can fall in that range, depending on the individual. Now, sterile sites I have listed here also. The sterile sites are those areas that are not in direct contact with the outside world in any way, unless there's a breach, right, in the system. Okay. Some people may say that blood you can find transient microbiota for short periods of time. That's a terminology thing. It's not necessarily, I wouldn't call it necessarily transient microbiota. Rather, we can have our blood can become contaminated, right, and not necessarily get an infection, right? Because if we have a nick or cut on the skin, we can get bacteria into the blood, but our immune system resolves the issue before it reaches the point of actually growing and multiplying in that environment. Say, for instance, the saliva. Saliva is, when it's produced, it is sterile, right? It's from the salivary glands, but as soon as it hits the mouth, it's contaminated immediately. Okay, and which is a big diverse group that is a very big diverse group population in the mouth. But anyways, you see the list is mostly internal organs, right? Now, where do we acquire this normal microbiota? So, the fetus in the womb is normally free of microorganisms, normally, right? Yes, we can have a infection state or disease state where there might be organisms that reach that point. But normally that's free of microorganisms. So the fetus then remains in a sterile environment until just before birth. Okay, once the water breaks, then you have the potential for microorganisms to move, but more critically, of course, is when the baby is actually moving through the birth canal. That's when they're picking up the starting of their normal microbiota, okay, which is primarily lactobacillus. Gut microbiota is usually established within 8 to 12 hours of birth. And that will shift over time based upon your dietary intake. It does make a difference as to whether an infant is born through the birth canal or whether they have a cesarean. Okay, so they've this picture just pulled off the internet, just kind of listed out. Vaginal birth, of course, is more the lactobacillus, which is what we're going to see in that location as the infant's traveling through, okay, being born. But cesarean section, they're missing that step. So then we see more along the lines of the normal skin bacteria, okay, the Staphylococcus and the Propium bacterium, those two. Now, again, the next, of course, is the gut, and now the gut does make a difference as to whether naturally like breastfed or bottle fed. Okay, there is a difference between as to what population you're going to see. In general, an infant who is breastfed will have a less diverse population, primarily of lactobacillus and the bifidobacterium. Those are the two. Okay, versus that infant that is bottle fed, not breast milk. Okay, bottle fed with not breast milk. If you bottle fed with breast milk, it's the same thing, right? But bottle fed without breast milk is another source. Then You have a more diverse population, and, but in the end, over time, right, all infants are going to transition from milk to solid foods, and as you transition to those solid foods, in the end, that diverse population is going to get there, even for the breast milk. Really, the additional that the breast milk fed are going to receive are the antibodies, right? That's the bigger thing that they're going to get that you're not going to see for those that are bottle fed. What other things are going to play a role in that gut population? Well, if they are have to take any antibiotics for any infections, okay, how they are fed, their own genetics is going to come into play, as well as the environment. Of course, infants and young toddlers are on the floor, right? And then things are always going in their mouths. So that's all contributing to that gut microbiota. Right after birth and during the first few months after birth, as that resident microbiota is being established, on the skin and other tissues outside of gut, you're really looking at anybody they interact with. Okay, mom, dad, family members, whether they be immediate with the kids or grandma and grandpa, anybody, any friends coming in to visit. If you decide to go take your child out early on in their life and you're going to the grocery store, anybody in those environments that look over and moo and coo over your infant, okay? Because spittle, of course, no matter, this is happening for everyone, and it's going to be transferring microorganisms. The doctors and nurses right at birth too. Yeah.
What's like a safe amount for like a baby to be exposed to bacteria so they have like a strong immune system versus like completely making everything so clean that if they do get sick it gets really bad? Like, would you say there's like a...
Well, you're always exposing, period, any infant to bacteria. I mean, just by the fact that they're breathing, they're being exposed. So you're talking about exposure to potential pathogens. That's a whole nother, ball game.
Right.
Obviously, our hope is not to expose them to pathogens too early in life, just because their immune system needs some time. And a growing infant is, a lot of their metabolism is involved with just growth, right? But you also don't want to wait either. So the only thing, mine is you just have to, you have to live life.
Yeah.
And the thing is you can't, it's not ideal to you want to take precautions, the same sort of precautions you would take for yourself, right? And go that route. Because if your infant is, first of all, right after birth, there are a lot of your IgG antibodies. The infant has your IgG antibodies. So anything you've been exposed to, they have circulating antibodies in their system. And that's going to last for a period of time. Okay, so it's going to last several months.
That's when you get the Tdap while you're pregnant. So
Correct.
you can have it.
Correct. Right, because you're responding and all those IgG are going to them, and so that covers them for a period of time. Absolutely. And then, so anyways, that's there, as well as, again, just if you're taking the normal precautions that you would take for yourself, because none of us are running around going, I want to catch that sick person over there to expose myself. No, we're taking the appropriate precautions to prevent ourselves. Does that mean we don't get sick? No, we still gonna get, likely at some point gonna run into and get sick. So, it's a, I think it's more of a personal preference. Years and years ago, you didn't bring your kid out into the population for like a month or several months, after birth. That's not the case anymore, So, and I don't think our population is any worse off for it, right? Because again, you're just going to, get your baby exposed sooner. But again, you're just taking the standard precautions, is what I'd say. And then as the child then moves into, you know, a toddler and starts walking, and so we're not looking at less and less hands on the ground and in the mouth, right? And then into young adult, puberty, young adult, etc. Your microbiota is still going to be changing. In fact, to give you an example, the microbiota of the stomach, that particular one, usually fully acquired if you're going to, depending upon if you get the particular microorganism, it's usually acquired and found in the bulk of the population by the age of 65. Prior to that, fewer, the numbers of people who have that particular bacteria is less and less, right? So you do acquire still even over the course of your life. Now, what is the impact of the normal microbiota? Well, first of all, is biological success. It is actually good to have that normal microbiota, both for the host, us as humans, as well as the microorganism, okay? So the microorganism benefits because we're providing a nice incubator and incoming food, and we're benefiting because, those microorganisms in the gut are breaking down the food, providing the nutrients that are able to be absorbed. Okay, some of these particular macromolecules are ones in which maybe we don't have the ability to break down. We rely on those microorganisms to break it down for us so that we can get those nutrients out of that food source. Also, what we call microbial antagonism. That is, if you have your normal microbiota, established, they are already present, right? So any pathogens coming in have to find a place where they can establish in an environment that's already established, okay? So think of it as, if you've been somebody who's moved around a lot, okay? I know when I was a kid, I was a Navy brat, so moved around a lot. You know this type of thing, right? You go into a new neighborhood. They have all the cliques already. They've been around each other for a long time. They don't want to let people in, okay? Well, this is the same kind of thing that's going on with these microorganisms. They've already established, I'm dominant in this area, this is my home, I'm not letting other people come in. In this case, people being other microorganisms. So that's what we call microbial antagonism, the fact that they're already established, so pathogens have a little more work to do to get established. As well as our normal microbiota actually helps stimulate our immune system, keeps our immune system active, right? So that's a good thing too that they provide. Now, can our normal microbiota become a problem? Absolutely, right? Absolutely. So when we look at our normal microbiota can become what we refer to as opportunistic pathogens. So we talk about pathogens. Pathogens fall into two categories. There's the true pathogens and the opportunistic pathogens. The true pathogens are your primary pathogens. They are capable of causing disease in healthy individuals with a normal immune, when I say that healthy individual with a normal immune immune defenses in place. These would be things like, say, the influenza virus, all your viruses. The plasmodium, which is the protozoan that causes malaria. Yersinia pestis, which is a bacteria that causes plague. These are true pathogens. If you have them growing in or on the body, you have a disease state, right? They're never found as part of the normal flora. That's just that they are never there. If as a person have them, you have the disease. That's a true pathogen. An opportunistic pathogen is again, that normal flora, that cause disease when the host defenses are compromised or when they become established in a part of the body that is not natural to them. Okay, so they're not considered a pathogen until they gain access to an area that they don't belong to, until they gain access to some of those sterile environments. Examples of that would be, say, the Pseudomonas. Okay, that's a soil microorganism. For the most part, we're not, a normal individual is not a healthy, intact immune system, is not going to be, it's not going to have a problem with Pseudomonas species unless it gains entry where it shouldn't be. Okay. Now, in a hospital setting, when we have that, we do see an increase in the number of diseases by this particular organism. Okay. Now, this is an example of an opportunistic pathogen that's not part of the normal flora. Okay. So it, depends upon the situation as to whether it be an opportunity, whether it's part of the normal flora or not. An opportunistic pathogen is, again, when it's an organism that is compromised and gotten established where it shouldn't be, but doesn't produce a disease for all people, right? The other example would be Candida albicans, that cause the yeast, right? A yeast infection. Now, besides being introduced into an unusual sterile environment, of course, we know that women can get yeast infections, right? In an environment that's quite natural for the yeast to be found. So in the vagina, that's part of the normal flora. They don't have to gain entrance to an unusually sterile location. That's always got microorganisms. But a woman can get the yeast infection when they have a change in pH, okay, a disruption in the pH in that environment. Or they're taking an antibiotic that kills off the bacteria, and then we got an overgrowth of the yeast, okay? So things such as changes in your normal microbiota due to maybe antibiotic being taken is possible for a means of an opportunistic infection to occur where it's not necessarily an unusual sterile environment to begin with. This is where they're normally found. Immune suppression can also be where we see opportunistic pathogens. Those individuals with say HIV and they have now got the acquired immune deficiency syndrome, right? At that point, now all of a sudden some of their normal microbiota can become opportunistic pathogens and a concern for them. Stressful conditions actually is linked, right? When you're stressed, your immune system is under stress, okay? So now, with a stressed immune system, that could be due to, studying for this class and taking this class as well as several other ones, right? Or unhealthy eating, not eating a good healthy diet, not getting enough sleep. Those are all things that can result in a stressful condition. But the thing with stressful conditions, it puts stress on the immune system. Immune system stress, now we're in a immune suppression, and you have a higher probability of getting sick. Okay, and we've probably all experienced that sometime in our life also. All right. Now, what are the manifestations of these diseases that can crop up by these pathogens, whether they be true pathogens or opportunistic pathogens? Well, we see these disease state as manifestations as symptoms, signs, and syndromes. So signs are something that is, I jumped. Let me start with symptoms because that's the first one on the list here. So symptoms are felt only by the patient, okay? So that's subjective. That would be things like pain, nausea, headache, sore throat, fatigue, right? Malaise, itching, cramps. These are all things that, when nobody can feel but that particular patient, right? And you generally, when asked about it, they give you a numbering system of 1 to 10, right? And it can be a big difference between people. One of my sons comes up to me with a big goose egg on his forehead. I'm like, holy crap, what happened? I don't know. I have something on my head. I'm like, come on. That had to have hurt when you fell and hit something that resulted in that, right? But again, how am I to judge his feeling of pain compared to my feeling of pain? One of my girls, on the other hand, barely gets knocked, and we hear about it for the next 2 weeks, right? Because it hurts so bad, and this and that and the other, right? So depends upon the individual. That's subjective. So generally with these, with the symptoms, we're looking at getting a baseline to start off, and then as we start treatments and et cetera, we then turn around and ask the same questions again, hoping that we see a change, right, in the numbers, because it's assigned by the particular individual. Signs are objective. They are observed or measured, but can be observed or measured by others. And so signs would be such things as swelling, rash, vomiting, diarrhea, fever, pus, anemia, increase or decrease in white blood cell count, heart rate, swollen lymph nodes. Those are all things that, you know, everyone can see the same resulting outcome, right? And then, so when we look at a particular disease, and when you go in to see the doctor, they're going to be asking about these signs and symptoms. And from these signs and symptoms, they can logically guess what the situation is, what might be happening for you based upon that, what we call the syndrome, okay, which is that combination of those two. Because they generally, each disease has its characteristic syndrome. Okay, is it foolproof? No, it's not foolproof, because any given disease, may in one person display, some slight variance compared to another. But in general, that's how when you go to the doctor and you give your complaints and your concerns to the doctor, they're saying, you got a viral infection, right, based upon that. Now, there are, of course, situations where an individual is asymptomatic or subclinical. Okay, and so these are infections that lack symptoms, but may still have some signs of an infection. May not. Okay, so it just depends upon the individual. And there is in table 15.1, by the way, I'm not going through it in my lecture, but there is a table there, that's table 15.1, that provides prefixes and suffixes that you should be kind of familiar with. I mean, especially if you're going into any of your allied health anyways. There's prefixes and suffixes that are related to diseases, okay, to help apply, say, if you're looking at a longer word and you're going, what does that mean? You can break it down by some of those prefixes and suffixes as to at least target organs or what type of infection it might be, okay? All right, so now the study of the disease itself, this is referred to as etiology. So etiology is the study of disease and the disease and what causes the disease. If it's an infectious disease, it's caused by a microorganism. In which case, then we want to identify the microorganism. And to do this, at this point in time, still today, the most common way is through Koch's postulates. So we talk about this actually in the first unit. Koch's postulates is one set up by Robert Koch, and he was the first one to link an organism with a disease, and that was Bacillus anthracis with anthrax. Excuse me, with anthrax. Okay, so he linked the two, and he made this link through this following process. It is a 4-step process where you observe diseased organisms and healthy organisms, and you look at the culture. You culture specimens from those hosts. Right? If it's a respiratory infection, you're probably going to be culturing sputum. If it is a digestive infection, so from the intestines, you're probably gonna take fecal matter, in order to try to identify, in a laboratory setting, what might be the causative agent. And really what we're doing is what's present in one population, the diseased individuals, that's not present in another population, that is the healthy individuals. Okay, what's there in one but not in the other, okay? And once you've identified that is found in one population, not the other, right? Then you move into the next stage, which is to grow in the laboratory setting, to characterize it, to try to identify it. If it's brand new, you might be adding it to the list, okay? But you need to grow it in the laboratory, and then from that you've grown in the laboratory, you're going to then move to the third step, which is then to inoculate a secondary host. Okay, and then that secondary host you're looking for, the intention, of course, would be that secondary host, if this truly was the pathogen, that secondary host would display the same signs and symptoms as the first host. Okay. And if they do, you're going to then take cultures from that host and try to, in the laboratory setting, grow those cultures, and then characterize that specimen. And the specimen should be identical, the characteristics, the profile that's set up with the biochemical test should be identical to that what you found in step 2. Now there are some ethical concerns, right? Slight ethical concerns, right? Especially if the microorganism causes death, right? Disease, sure, that's also ethically, concerning, but if the outcome is death, then, that whole, who do we inoculate for a secondary host is not, clear. So we generally use another animal, the mice, rats, or rabbits, sometimes depending, cats or monkeys, depending upon the particular pathogen and whether they grow. The thing is that if your secondary host is a different species, you might not see the same reaction or disease state in that secondary host, right? So then that can become problematic. Okay, but we're trying to find, do we have that same reaction with that secondary host? Another problem that we run into is some pathogens we have difficulty culturing in the laboratory setting. Okay, so finding the right media mix that they can survive in a outside of the body is not always easy. Like in our class, when we head over to lab, everything grows for us, right? Sometimes more things than we wanted to grow for us. But when we are looking for these situations, in fact, and even with your normal microbiota, there is still a question as to whether we've even identified all of the normal microbiota that are out there, the bacteria that inhabit our body. Because we can't grow them in the laboratory setting, okay, to do the characterization of them. Another difficulty we run into is the situation where the disease is caused by a combination of pathogens, right? It's kind of the perfect storm type of situation where it's not caused by one organism, it's caused because you had this organism, or maybe this organism, and the other organism, right? So it's because you had maybe a two or more organisms, and the combination resulted in this disease state. That's going to be problematic for this process. Okay. Another thing that's going to be problematic for this process is if the disease is the cause of the disease is normal flora, microbiota. I told you I'd switch. Normal microbiota. Because you're going to find that organism in your diseased population and healthy population. And what was the first thing we wanted to do? Find an organism in just the diseased, not in the healthy. But normal flora is found in both. Okay, so your opportunistic pathogens that are part of the normal flora sometimes are difficult to associate with a particular disease because it's hard to identify it. All right. Covered. Butchered that slide good enough. So, then we get to our chain of infection. This is just a picture that I found online that kind of gives you some of the parts and pieces. The chain of infection, though, really is contact, infection, disease, continuum. Okay, and that repeats and repeats and repeats. For that to happen, the contact, right, would be how do those organisms get around? What is that mode of transmission? So we'll be this unit will be module will be going through that later. What is the mode of transmission? How does it get passed from one individual to the next, right? And once it does get past one individual to the next, it's got to get into the system. We have lots of mechanisms in place to prevent ourselves, to prevent illness, to prevent these microorganisms from establishing an infection in us. And the key is the organism has to enter through the portal of entry. Okay? Whatever that might be. It's specific for the organism. Okay? Or virus if we're talking about that. And then, of course, what's going to also come into play would be susceptibility of the host. So there's some things that are going to contribute to not just the fact that the organism comes into contact with the portal of entry, but there are some additional things that are going to come into play, such as children, whatever age they fall into, right, tend to be more susceptible. It has to do with their immune system. Elderly tend to be more susceptible. Also has to do with their immune system. Okay? People with weakened immune system, unimmunized people, right? If somebody is pregnant, that might play a role. Okay, and then some pathogens it doesn't matter. They don't have a particular group that's more susceptible than the others. Are kind of non-discriminate. You know, just anybody and everybody. Okay, that depends upon the pathogen to some extent too. So then we do have those particular pathogens, such as bacteria, your viruses, protozoans that might cause disease, your parasitic worms, your fungi, right? So just broad range of microorganisms that are gaining entry. Now, where are they generally found? Well, while they're in a sick person, we know where they're found. They're found in the sick person. But, you know, when they're not in the sick person, right, where are they found? That is referred to as the reservoir, where the germs live naturally, normally. And then we also then have, eventually, if somebody is sick, we're also going to get, the germs have to eventually get out to be able to move to the next host, right? To continue this process of, we have to come in contact, we get the infection state, the organism grows, we get the disease, and then they have to leave so that they can move to the next host and repeat the process. Our body surfaces are constantly being exposed to microorganisms. Okay? Now, that may or may not lead to an infection. So we need to make sure we're clear on what is an infection. Contamination is when we come in contact, right? We become contaminated all the time. That doesn't mean we're getting sick from those pathogens. Say, for instance, COVID. What we just went through a big pandemic, you know, that probably altered a lot of people's lives, whether they got sick or not. Okay, but in this case, fomites is coming in contact with fomites that might have, or in the mode of transmission, close proximity to a person, right, as they're speaking. So contact that throughout. Doesn't mean that we are going to get infected. So you help an individual with COVID, you get it on your hands. Well, the portal of entry is through the respiratory tract, which has access through the eyes too, right? The mouth, the nose, and the eyes.
In the ear.
And the ears, yes. And so you get it on your hands, you've contaminated yourself. That doesn't mean you're going to get sick. It doesn't mean you're going to get an infection. It's only then if you turn around and touch the areas that are the point of contact, or the point of entry, excuse me, not contact, the point of entry, do you have a higher probability of getting sick. That doesn't mean definitely you will, though. There is still what we call the infectious dose, a certain number that you have to come into contact for you to get the infection, which may or may not be met. Okay, and then of course you have to have that the organism must adhere. So there's a lot of steps involved before you actually potentially will get this infection. And the infection is when is defined as that time frame when the organism is colonizing that host. So actually growing in the host. Infection does not mean disease. So you can have an infection and never reach the disease level. Okay. The infection is the invasion and establishment in the host. The types of infections you can have would be a local infection, say just maybe one place on the skin, or in the mouth, wherever, right? It can be a focal infection, and focal infection is where it starts out as local and then moves to a secondary site. Okay, this is pretty common with dental caries. When you have tooth decay, and if you don't get treated in time, the decay is sufficient enough that it gains access to the circulatory system, right? And then it can enter the circulatory system where many of these bacteria then move through the circulatory system and attach to an area, to the heart area. Okay, now all of a sudden you got endocarditis. Sepsis, no. Endocarditis is that, an infection of the heart. Sepsis is bacteria present and growing in the blood. Is a definition I'm eventually getting to, but yes. It is in my list of definitions that are in this unit, or this module. But anyways though, and then we have the systemic infection. The systemic infection is when it's throughout the entire body. One example of systemic infection would be sepsis, right? Because it's now circulating throughout, the organism is circulating through many different organs of the body, or throughout the entire body. Of course, we also have the primary infection and the secondary infection. I already mentioned those. The primary is the first one, the first microorganism that resulted in an infection, and then that caused opportunity for some other organism now to cause it, to overgrow and become an infection itself too, right? Now, these infections, all these infections, we move into from infection to disease state when we have that pathogen is altering normal body functions. Okay, so now you have a disease is where you're going to have that pathological state where there's damage or disruption of tissues or organs, right? Some normal processes are being affected. Now you've moved into what we term as a disease. Okay, the disease state, another term for that would be morbidity. If the disease results in death, that is mortality. Those terms are going to come up with epidemiology. Okay, so that's which is covered in this module here. Now, the diseases specifically that we are focusing on would be the infectious diseases, right? Diseases that are caused by microorganisms. There are many other diseases, non-infectious diseases, that are not going to obviously follow this chain of infection because they are caused by other reasons, say hereditary, okay? maybe unknown causes. That's also an option. So there's a whole bunch of terms that they have a table of terms that talk about that are for non-infectious diseases, those that are not caused by microorganisms. Infectious diseases are the ones we're focusing on because those are caused by microorganisms. And infectious diseases can be classified as communicable diseases, which means they transfer from person to person, right? In the communicable, we have a subclass of contagious communicable infections. So contagious infections would be, or diseases would be ones that transfer very easily from one host to the next. We also have then our non-communicable diseases. Non-communicable diseases are not transferred from host to host. Okay, you always are getting it from the environment, or from another animal. If you are getting a disease from that's normally found in animals, that's being moved, that's transferring over to us, so now we're sick from that same pathogen, those are your zoonotic diseases. Okay, your zoonotic diseases, and we'll talk about that too for your transmission later as part of that transmission process. And then your nosocomial diseases are those, or oftentimes now termed as healthcare associated infections or diseases, depending upon which it falls under as far as a class. Those are the infections or diseases that are acquired from the hospital setting. Okay. All right, so what are our reservoirs? Okay, our reservoirs are the location where the pathogen is going to survive and exist outside of the host. Okay, and there are three main types of reservoirs. We have our animal reservoirs, human carriers, and non-living reservoirs. Human carriers obviously could be classified as a subclass of animal reservoirs, right? Because officially we are animals. But when they say animal reservoirs, they're generally talking about other animals aside from humans, all right? And so these are also the diseases that are acquired through these sort of reservoirs are referred to as our zoonotic diseases. Okay, so zoonotic diseases are diseases that we're a secondary host. We're not the primary host. So this organism moves through a different population normally. And occasionally, because we interact with that population of animals, we get sick. For zoonotic diseases, we don't transfer to other people usually. These are not communicable diseases usually. Okay? The next person who gets sick got it from another animal. How do we acquire the zoonotic diseases? They can be acquired different routes. Some is through direct contact with the animal and its wastes. Hantavirus is one that's seen over in the southwest with arid conditions. It's from the mice that live in the desert, and they're the primary. They urinate, very arid conditions, and then you see it pop up, the disease pop up during the Santa Ana winds then take that dried urine and bring it in the air, and people breathe it in, and it causes a respiratory disease for us. Yeah. So sometimes, right, it could be contact with the waste products of the organism, of that animal. Other times, we have our domesticated, we have pets, domesticated cats and dogs that we're petting, and sometimes we can get transferred that way. Another is through eating of animals. Okay, if the food source the meat product has not been properly cooked. Okay, reaching the appropriate, it's an easy way to fix this problem, is to make sure that the temperatures, the required temperatures are met when you're cooking that particular meat product. Okay? And then the other would be through blood-sucking arthropods. Fleas, ticks, mosquitoes. Okay, would be your three most common. Again, we are generally dead-end hosts, so we are not passing it on to other people. People are getting it from interaction. One of those three mechanisms in the environment. It is very difficult to eradicate these type of diseases, those that fall in the animal reservoir, because of the fact that it, like for instance, one example would be rabies. Lissa virus is the cause of rabies, and it's found in the population. It can be found in the population for dogs. Cats can get it too, right? And so domesticated cats and dogs, we have the requirement in Florida they be vaccinated, right, yearly. Ferrets also, okay? And we have control over that because these are our pets. We take them in, get them vaccinated. Prevents the likelihood that they're going to get the disease. And we interact with them, so we're less likely to get it. But there are feral cats out there, there are feral dogs out there. We cannot get the whole population. On top of which, that particular virus, it lives in its reservoirs can be bats, skunks, foxes, okay, armadillos, pretty large range, almost any mammal, okay, is susceptible. So the likelihood that we can get out there and vaccinate all of these feral animals is not going to happen. Therefore, it is something that's going to be present in the population as a possibility that comes up, right? Other examples, malaria. So Plasmodium, malaria is transferred from one host to another by the mosquito. Its primary host is actually monkeys. Bacillus anthracis, primary host, that is the cause of anthrax. Its primary host is herding animals. So herding communities, goats, cows, that type of thing. It's pretty commonly found in that. As a potential disease that pops up in those type of communities. Salmonellosis is caused by Salmonella species. Salmonella is very common in birds, rodents, and reptiles. Okay, that's their primary. We get it due to, say, for instance, when undercooked eggs, or the egg shell might have some on the outside, and of course we prep our egg, that's same with our meat, same way, is we prep it on maybe in a counter, but we know we don't properly disinfect before we move to the next food product that we're prepping, right? And maybe the next food product happens to be a salad, which isn't gonna get cooked. So you might have cooked the egg before you ate it, but you didn't cook the other item, whatever it might be. Toxoplasma gondii is primary host is cats, causes toxoplasmosis. That one's a fun one actually, I think. Toxoplasmosis transfers between the cat and the mouse, okay? And what it does is it causes a change in the brain so that the mouse gets an attitude, thinks that they can get away with things, right? It reduces their inhibition, which actually makes it easier for the cat to catch it. Cat eats the mouse, cat gets it, right? So it goes back and forth that way. So interesting enough, we can get it too. And if you're a watcher of many of your medical shows have had it, Law and Order has had it, where they'll have, because it does the same thing to us. Only difference is, it's that loss of inhibition usually means the person becomes promiscuous. Okay, and so they start, all of a sudden somebody who normally wouldn't be running around sexually active with all sorts of different people, all of a sudden becomes that way. and so it's been it's an interesting one, and it is actually the organism, the pathogen that is doing this. And there's a reason, like I said, the reason is so it makes it easier for the pathogen to transfer from the mouse to the cat. I think it's interesting. I think all sorts of things are interesting. Human carriers. Human carriers, in the case of human carriers, here we're talking about somebody can be an asymptomatic carrier. In fact, salmonella actually happens to be one of those examples. An individual can be a carrier for salmonella and infect other individuals with that disease, an intestinal disease. One of the early on human carriers for salmonella, they gave the name of Typhoid Mary. She was an individual, and this was back in the early 1900s. She was an Irish immigrant to the U.S., and she worked as a cook. Keeping in mind that during this time, we're still talking about an early time where, personal hygiene was still being learned about. The connection that microorganisms cause disease had been discovered, but still new. And she would cook for the family she's working for, and the family would get sick with typhoid, and people would die. The family, individuals in the family would die. She would then move on to another household. They'd get sick. And so what they found was, as she started, as she was being observed, they said that groups came in and said, "You can't be a cook anymore. Okay, you have to find other work because we think you're getting people sick." And her stance was, "I've never had it, so it can't be because of me, right?" Other option for females at the time was cleaning. Cleaning doesn't get paid as much as cooking. So she would just move out of that area into a whole new area and get hired on to cook again. And people get sick. They actually forced quarantined her for a portion of time, and because she was adamant that she was not getting people sick. So she was one of the first to where they were able to demonstrate that her as a human carrier for salmonella. We see this. Streptococcus pyogenes, some people are carriers for this. When I took my medical micro in my undergrad, they had us doing a throat culture. And I discovered at that time, I happened to come up with the organism of the throat culture that I worked with and identified happened to be Streptococcus pyogenes. Okay? This means that I'm more likely to pass it on to other people, potentially, but I'm not getting it sick by it. Right? Now, can I get sick by it? Absolutely. It is an opportunistic pathogen, so at some point or other I could. Now, since that time, I've taken antibiotics numerous times in my life, so has that shifted? Do I still have it? I don't know. Okay? It may have changed. But at that time, yes. But I was not sick during when I was taking that class. So I would have been what we would consider a carrier. Right? So the individual does not usually show any signs and symptoms of this, so they're asymptomatic. Now, they can eventually develop the illness, or they may never. It depends. Some individuals are what we call the convalescent carriers. So for a period of time after you've been sick, okay, so they've already recovered. They're in what we call the convalescent portion of the disease state, so they're recovering at this point, no signs and symptoms, and yet they still have enough organisms present to be able to pass it on to somebody else. Okay? Convalescent carriers are it's usually a shorter period of time, but it could be, like salmonella is one that somebody could be a convalescent carrier, and that can last for over a year. Okay? They already have recovered, but they're still carrying a little bit of residual, okay, before it gets finally cleared. And then our non-living reservoirs would be your soil and water. Okay, the soil and water as a reservoir, a lot of times it is often due to contamination of the water supply or the soil by feces or urine. Okay, but they're able to stay in that environment, survive in that environment in the interim before being consumed, right? And you say, well, water, yeah, sure, we consume water. We don't go around eating soil. Well, yes and no. We do eat plants that are coming from the soil, right? And so it's possible if they're not properly washed, there's still some residual present, and therefore we're still consuming. So oftentimes foods are linked with our non-living reservoir when we talk about food sources, okay? Even though everyone knows plants are living, right? Just they don't get to move around, but they are living. They still though fall under the non-living reservoir.
Well, they should be dead if you're getting sick from them.
They should be dead.
Yeah. If you're
Well,
eating
based
plants,
on
they're
that
dead.
theory, whoever, when last time you got sick, whoever transferred it to you should be dead, because they
No.
gave you an illness.
I'm
Right.
saying, if you eat the plant, you already took it from the living source. So that.
Well, you're digesting it. I'm gonna stop here. I'll start up at this point on Monday, okay? So we'll go ahead and start in the lab at 7:35.